Well, welcome to the first segment for Chapter 14. So this is really the first chapter regarding genetics. So we're going to talk about Mendel, of course, because he was the one who really did a lot of studies and discovered a lot regarding inheritance. So let's go ahead and start with some vocabulary that's kind of necessary. All right, so these are some useful vocabulary words. However, of course, all the way through biology, there are a lot of vocabulary words which need to be learned. But these are the most important for this particular chapter. Now, the first one is allele. So allele is basically an alternative form of a gene or a form of a gene, right? So often the alleles are going to be represented by letters. And sometimes the letter is going to be, I guess, chosen based on whatever character or trait in which the allele codes for. So here I have an example. Okay, so these are two forms of an allele. We have a capital A and a lowercase a. So this will kind of become important later on. And the difference between the capital and the lowercase regarding genetics is that when an allele is displayed as a capital letter, it is considered what's called dominant. And when it's lowercase, it's considered what's called recessive. Dominance and recessiveness really has to do with how the gene is expressed, right? So what you actually see from the products of the gene. And we'll kind of expand on that later. And you may already be familiar with that. Now, another term is homozygous. So when you put two alleles together, remember, most organisms are diploid, meaning that they have two copies of each chromosome. So then they must have two alleles for each gene. So when you put the two alleles together, when they're identical, meaning, let's say you have two capital A's or two dominant alleles or two recessive alleles, that is called homozygous, meaning the same, and we're referring to zygote or zygous when you're bringing the two alleles together after fertilization. That means they're the same. Okay. Another term for this is called true breeding. So you might see this phrase in the future. True breeding means that the genotype for that organism is homozygous. Now, that individual could be homozygous dominant or homozygous recessive. Now, as opposed to homozygous, there's heterozygous. Hetero meaning different. So this means that this individual has two different alleles or two different forms of a gene. So this is shown like this, right? So one of them's capitalized here and one of them's lowercase. Okay, so referring to the difference between homozygous and heterozygous and dominance and recessiveness, when an allele is dominant and the alternate allele for that gene is recessive, and an individual has at least one copy of the dominant allele, they will display that dominant trait. Now, we're going to kind of talk about this as we go through, and if you're not already familiar with this, you will become familiar with this. Now, here's an example. Now, before we move into the example, we should talk about the difference between genotype and phenotype. Now, genotype is really the genetic makeup. So the genetic makeup is often referred to as alleles, and what alleles are, is it the dominant allele or is it the recessive allele? Does this individual have two of the dominant alleles, two of the recessive alleles, or one of each? Okay, so genotypes, these are examples of genotypes right here. Okay, so capital A, lowercase a, and then capital lowercase. Okay, so these are all genotypes. Each of the letters is an allele. Now, what the phenotype is what you see from the genotype. For instance, if capital A represented brown eyes and lowercase a represented blue eyes, okay, an individual, in order to express blue eyes, must have, since blue eyes is recessive here, must have both of the recessive alleles, because if the individual has one of each, because the capital A is dominant or the brown eyes are dominant, that's what you're actually going to see. Okay. Now, the allele for the recessive trait is still present in that individual, but what's expressed is the brown eyes. Okay. And of course, the homozygous dominant individual is going to have brown eyes because they have two copies of each, two copies of the dominant allele. Now, let's kind of look at an example, which is kind of related to an example that we've just been discussing all throughout the semester, and that's the sickle cell disease. Okay, so if we look at the genotype of an individual, well, okay, so the gene for hemoglobin is on chromosome 11. Now, we have hemoglobin A and hemoglobin S here. So hemoglobin A really represents the normal hemoglobin. And then the hemoglobin superscript lowercase s represents the sickle cell allele. And what this is it's a mutation at the nucleotide level, which results in the change in an amino acid. Okay, so the chromosome here represents the nucleotide level. Okay, so here we have the nucleotides, which make up the chromosomes, and now we have the red area, which represents a gene for hemoglobin, and this is the mutant gene right here. Okay, so this is the one that has that mutation in the nucleotide sequence. Okay, so let's say an individual has one normal and one sickle cell. That would be a heterozygous individual, right? Now, what's expressed from this is what the phenotype is. Okay, so if we looked at it at the amino acid level, okay, so what's actually expressed when the protein is made? The normal one has a glutamic acid in the position where the mutation is in the sickle cell allele. However, because the mutation is in the sickle cell allele, this results in an amino acid change to valine. Okay, so what we see from these mutations, okay, what's expressed in the normal hemoglobin, it's a glutamic acid. What's expressed in the sickle cell hemoglobin, that's a valine. Okay, so that's one example of the difference between the genotype and a phenotype. You will see many examples during the next 2 to 3 weeks. All right, so let's look at Mendel's experimental. He used a quantitative approach while studying garden peas. Now, he actually discovered the principles of heredity. So how are things inherited using these garden peas and breeding them? Now, he planned these experiments very carefully. Now, some advantages of using pea plants, right, for genetics are the following. First of all, there are many different varieties of them with distinct heritable features. So these features can be easily visualized, so they can be studied easily. So these distinct heritable features are called characters. An example of a character might be the flower color or the pea shape. And character is really a broad term, okay, which includes many traits. Okay, so if we were going to talk about flower color, the character flower color, the traits that are associated with it are the individual colors. So we're going to see that the flowers can be purple or white, okay? So purple and white flowers are the traits that are associated with flower color as a character. Another example might be, let's say, a character eye color. Okay, so in humans, eye color has several traits associated with it. And this might be blue eyes, might be brown eyes, might be green eyes, right? And then there's different levels of eye color, huh? So there's quite a bit of variation in eye color. That's because eye color is not expressed due to a single gene. Okay, so there's multiple genes that contribute to eye color. And we'll talk about that in the second segment. Now, the next thing is the mating can be easily controlled. Right? So you can control, you grow the plants in separate pots or you keep them separated. They're easily manipulated. Another thing associated with that is the floral organs can be easily separated, right? So you can separate the males and females, okay? And then pollinate when you want to. And then, of course, cross-pollination involves fertilization between separate plants as opposed to self-pollination. Okay, so self-pollination is more like inbreeding plants. You want to cross-pollinate. Okay, so you want to pollinate between different plants. And it's relatively easily done, right? So you take the pollen, you easily brush it onto, the pollen's a male part, you brush it onto the female part, and then fertilization can occur. Okay, so these are 4 advantages of using the pea plants for the study of genetics and heredity. Now, Mendel noticed a trend. Okay, so what he did first was, so there's certain generations, and it's important to know how these breeding experiments are done. However, they're done a little bit easier nowadays. However, he bred these plants. Okay, so we have this parental generation. He took the parental generation, which were each true breeding for each of the 2 different colors. All right, so we have purple flowers and white flowers. And what true breeding, remember, means is that the purple flowers are homozygous dominant for purple, and the white flowers are homozygous recessive, and they have to be recessive because the white flowers are the white color is recessive, so they must have both of the recessive alleles. So these are what's called contrasting true breeding white and purple flowers from pea plants. Okay, so that's the parental generation. When you the F1 generation. And what he noticed was the F1 generation were all purple, even though one of the parents was white. So what happened, right? So how are you going to determine what happened? The best thing to do would be to cross the F1 generation. So take 2 of the F1 generation. So 2 of those purple flowers from this generation right here, and then cross those and see what you get. Right? So see what happens. That brings about the evolution. What he discovered was about a 3-to-1 ratio of purple to white. And of course, he repeated this, right? So he saw this, and then you're going to repeat it. You should always repeat things in science. And he noticed the same trend, right? So in the F2 generation, he noticed that there were basically 75% of the purple flowers and 25% of the white flowers. Now, he actually noticed this with all the other traits, right? So he observed the same pattern, this about 3-to-1 ratio when he already studied flower color, flower position, right? So where are the flowers located along the stem, the seed color, the seed shape, the pod color, and the length of the stem. All of these traits, he saw that same 3-to-1 ratio. That would indicate that that's pretty important, right? I mean, if that's happening the same way and it's just not completely random as to how they turn out, there must be something significant about what is creating this trend. How is it transmitted from one generation to the next? Now, what Mendel called this that's being transmitted from one generation to the next is a heritable factor. Okay, we now call a heritable factor a gene. Okay, so we call them genes, he called them heritable factors. Remember, this is before we knew about genes. Now it's almost taken for granted. We all know what a gene is. Now, he developed a model, basically a hypothesis to explain this 3-to-1 ratio of phenotypes that he saw. He described it as, of course, an inheritance pattern. Now, there are 4 related concepts that make this model up. Okay, so the first one states that alternative versions of the genes or alleles account for these variations in the inherited characters. This is what we see when we see the 2 different purple or the 2 different color flowers. We see purple. There has to be some alternative version of this heritable factor or gene, right? So that represents these colors. Again, these alternative versions are called alleles now. And each of them resides at a specific position on a chromosome or locus. Okay, so a locus is a position on the chromosome where each of the genes that have their alleles are positioned. Okay, so locus, you can kind of think of it like location. So here we have homologous chromosomes. Remember, you really get one from mom and one from dad, but remember they're kind of a mixture of your chromosomes are a mixture of mom and dad. However, you're going to end up with two alleles for one particular character. And here we're talking about the color of the flower. Okay, so this particular individual with these two chromosomes is a heterozygote because they have an allele for purple flowers and a corresponding allele for white flowers on the other of the two homologous chromosomes, right? So this is the locus, right? So this is the location in which that allele is positioned, and they're positioned in the same spot, or they should be positioned in the same spot on each. The second one, for each character, such as flower color, an organism inherits two alleles, one from each parent. Right. So he basically deduced this without really knowing the role of chromosomes or the function of the chromosomes, right? So they don't even know how this is actually done yet, but Mendel actually kind of deduced this from his studies. Right, so the two alleles one allele comes from mom and one allele comes from dad. They may be identical or they may be different. Okay, so this is a case where they're identical, right? So one of these came from mom and one of these came from dad. Okay, so both mom and dad have the same form of the allele. Okay, and then here, same thing. One of them comes from mom, one of them comes from dad. Again, they're the same form, but they're different than this one, right? So this is the homozygous dominant individual. This is a homozygous recessive individual. And then, of course, they could be different, right? So this is an individual that has both of them. And since purple is the dominant color, that's going to be seen as the phenotype when an individual is heterozygous. Now the third one. If the two alleles at the locus differ, then one, the dominant one, determines the organism's appearance. That's pretty much what I just said on the previous slide. Right? So, and then of course the other one, the recessive allele, doesn't have any effect on the appearance, but it's still there, and it can come out later during segregation of alleles and fertilization of the next generation. So in the flower color example, remember the F1 plants were all purple. Okay, and it's because every one of them had at least one copy of that dominant allele, so then they're going to display that purple color. So here you can see, remember all the F1 generation is heterozygote. But because each of the heterozygotes have one of the dominant alleles, then they're going to show or display that purple color. So what this third one is basically saying is that this dominant allele is going to determine the appearance of the organism. And it does, right? So the dominant allele, because there's one dominant and one recessive here, the dominant is going to be what the appearance is, right? So you're going to see the purple color. If an individual has two recessive, then they will show the recessive trait. And in this case, it's white flowers. The last one says, this is kind of related to what's called the law of segregation. The two alleles for each of the heritable characters, they separate or segregate during gamete formation. Okay, so remember meiosis from the previous chapter? Okay, the formation of gametes ultimately separates the two alleles. And remember, you were already introduced to the law of segregation. And of course, they're going to end up in different gametes. So an egg or sperm is only going to get one of the two alleles of the organism that produced the gametes. Okay, so if it's a female and that individual is homozygous dominant, then all of the eggs are going to have the capital B in this case. Okay, let's say the male, they were recessive, right? So they show the recessive traits. So all the sperm are going to have the lowercase b. Well, when they come together, this is fertilization right here. They create a zygote, and the zygote is going to be heterozygous in this case, right? So if the female was heterozygote, then 50% of her eggs are going to be the dominant allele, and 50% will be the recessive. And the same thing would happen with the sperm, right? So it'll be about 50% that would be dominant, 50% that would be recessive if these individuals were heterozygotes. Okay, so again, all of this results from the separation of the homologous chromosomes during meiosis or gamete formation. Okay, in summary, I discussed some of these terms. We talked about allele and described the difference between homozygous and heterozygous, the difference between genotype and phenotype, and what a locus is. Furthermore, we talked about Mendel's model and basically the hypothesis he came up with to describe what he observed when he studied the peas with that 3-to-1 phenotypic ratio.